Hi, my name is Alex. I work in the Ruberti Lab and I'm a master's student. And so our lab focuses on engineering of the extracellular matrix. And the extracellular matrix is proteins and molecules that exist outside of your cell. And so one protein that we're specifically interested in is collagen. And collagen is the principal load-bearing molecule in vertebrate animals. Collagen is also an interesting molecule because it's found everywhere in your body, from your skin to your tendons, even in your eyeballs. Also, collagen has been shown to be mechanosensitive, which means that it can adapt and change under load. And so when load is applied or when forces are applied, we've actually demonstrated that collagen can assemble and form fibrillar structures, which is really interesting. So now I want to show you some of the data that I'm collecting. So I'm doing long-term live cell dynamic imaging at high resolution. And so basically what that is I take a high resolution photo every 5 seconds for several hours to look at the dynamics of how cells are actually moving. And so right on the microscope, those are the cells. They're sitting in a temperature controlled dish so they can stay in a constant environment and temperature during the imaging process. And over here you can see the video, and this is the data that I'm collecting. And so as some background of what you're actually looking at, these cells are fibroblasts, which are cells that are known to produce a lot of collagen. And these are actually corneal fibroblasts. And so the cornea is the clear part at the front of your eye. And the reason we use corneal cells is because they are shown to create highly organized collagen. And so how I obtain these cells is I receive donor corneas, I dissect them. And so since we're engineers, we think about this from an engineer's perspective. So how can we use collagen and our knowledge of collagen assembly as a tool for therapeutics? And what's really great about collagen is since it's everywhere in your body, if we can really control its assembly, we can theoretically create therapeutics for everywhere in the body, such as wound healing, such as tissue engineering. So I first learned about Professor Ruberti's research my freshman year of undergrad at Northeastern. And I was really interested that he focuses on the effect that forces have on cells and tissue development, because previously I had only really learned about the effect of chemicals on cells. So I thought this was a really cool perspective, and I wanted to do more research into this. And so I actually joined this lab as a freshman with very little research experience. And so this lab has a great culture where they really took me in and made me feel like a member of the lab, and I got to do a lot of independent research projects, and I stayed here for my master's, and this is my master's thesis. And being a part of this lab has really helped me blossom into the researcher that I am and helped me find my path forward after graduation. And so I really love the tissue engineering research that I'm doing here. And in the future, I hope to be on the cutting edge of tissue engineering and translational medicine. And so after my master's, I hope to pursue an MD PhD, continuing this tissue engineering research.